Hello, everyone, and welcome to our lecture on the basis of taxonomy. This is chapter 10 in your textbook. And in this chapter, in this lecture, we're just going to cover the basics and history of taxonomy, and then how we do taxonomic classifications now, and then also how to read a phylogenetic tree and what does it mean when you're looking at these phylogenetic trees. So before we get into the history of taxonomy, I want to begin by saying that taxonomy is arbitrary and dynamic. So in your textbook, you'll notice that they give many examples of different methods that different scientists will use to identify a species and understand its relatedness to other species. And every scientist has their rationale for why they feel that their system is better than others, or why some systems need tweaking in order to be valid. So it's a bit arbitrary, but it's also dynamic. So as we get access to more information and more technology over time, the systems that we use, we have to tweak them. So like the advent of molecular technology, and then also, identifying that evolution exists has changed how we do taxonomy over time. So overall, taxonomy is extremely important and it's very effective, and it's key for us to understand the world around us and how everything kind of relates. But it does change over time. Like many things in science, it's dynamic, it's changing. So just keep that in mind as we're going through this PowerPoint. So historically, we as humans grouped animals based on their morphological similarities. So if you were a scientist and you were traveling through some territory and you found an organism that you didn't recognize, it was an unknown, you would catch that organism or take a detailed note of it in a very detailed picture, and then bring it with you back to a museum. And at the museum, they had what we call type strains or type species. And so these were samples of organisms that were supposed to be the ideal type of a particular species. And so you would compare your unknown to these various type species and try to figure out what your unknown sample is most closely related to. This was all visually based, and there were it was effective at that time. This is all pre-evolution. So this was just people trying to figure out, based on what I can see, how are these things related? The father of modern taxonomy, as we know it today, is Carolus Linnaeus, and he's considered the father of taxonomy because he developed a couple of different systems that we use today. Now, these systems have been modified, but they're still the basis of taxonomy, no matter what kind of system you're using. So the first thing is he developed a hierarchical system, so where you have species, which is the most specific, are different species are grouped into a genera, which is larger, and then genera are grouped into an order and then to a class, right? So he came up with this hierarchical system of classification, and all these groups are called taxa, or you'll hear me say taxa or taxon throughout this presentation, and just that it just basically means a grouping of organisms. So he came up with these different taxa that are organized in a hierarchical system. He also came up with the binomial nomenclature system of naming species. And so we'll talk a little bit more about that in our next slide. But this is a system that we still use to this day. So some problems with this system of just comparing based on what you can see are homologous and analogous characters can arise. And this can give you a false interpretation of how close two organisms are related, two species are related. So looking at homologous features, these are characteristics that are derived from a common ancestor. So all of the species that have this feature all have this feature because they share a common ancestor. So when we're looking at four-limb structures, you can see there's common features here, right? A cat, a human, whales, and bats all have similar structures to how their forelimbs are designed. It's because they all share a common ancestor. But then you have something on the other hand that are analogous traits. And so these characteristics are based on common selective pressures that organisms faced. So there was a common shared selective pressure, and so they evolved similar characteristics. But when you're looking at them just that for morphological similarity, they look the same. And so this can lead you to make improper interpretations of the relatedness between species. So the example here that we have in this image are comparing a shark, a penguin, and a dolphin, right? We know today that a shark, a penguin, and a dolphin are not related. One's a shark, one's a bird, one's a mammal, right? They are related, but far, far, far back. They are not closely related to one another. But if you were out in the field and you're looking at them, you may misinterpret their relationship because they all have fins, but their fins are actually a result of selective pressure. So they're analogous traits, not because of a common ancestor. And you can even see it in the bone structures of these fins, right? So even just comparing a shark and a penguin, their bone structure is very, very different because they did not derive from a common ancestor. They derived from just the same selective pressure out there in their environments. So in an effort to figure out maybe a more accurate way of looking at the relationship between species, they're switched from a focus of strictly morphological similarities to actually evolutionary relationships between organisms. And so this study of classifying species based on evolutionary history is called systematics. So looking more closely at the taxonomic classifications that we use for species, this is, like I mentioned, is based on Carolus Linnaeus's initial kind of hierarchical structure, but we've expanded upon it since. And so there are 7 major classifications. There's domain, kingdom, phylum, class, order, family, genus, and species. Well, yeah, thank you. Sorry, 7. I miscounted my hands. So there's 7 classifications, and with the largest being the domains. So the 3 domains of life are eukarya, bacteria, and archaea. And all life fits within one of these 3 categories. Back in Carolus Linnaeus's time, he didn't have access to RNA sequencing, which is how we came up with these 3 distinct domains. So his classification didn't have domain. This is something that's been added since due to our RNA sequence analysis. But if we look at this image here, we have our bacteria, archaea, and our eukarya organized on a phylogenetic tree. And this down here is our common ancestor of all life, so our first kind of cell. And the two groupings that are considered prokaryotic are the bacteria and our archaea. So prokaryote. Sorry for my poor handwriting. I'm trying to write on a surface that's in the wrong direction. So then we have our eukarya. And when you're looking at archaea and bacteria underneath a microscope, they actually look very, very similar to one another. And you would mistakenly think that, okay, well, they're the same or at least closely related. But in actuality, archaea are more closely related to eukarya than they are to bacteria. So an archaea are more closely related to us than they are to, say, Pseudomonas aeruginosa, which is a bacteria, which is, to me, quite interesting. So remember, prokarya means they do not have a nucleus. Their chromosome is not nuclear bound. And then eukarya have a nucleus, true nucleus eukarya. Okay, and so there are 7 basic groups, like I mentioned. Every living animal, every living thing on life must fit within somewhere in these categories. But depending on what kind of species you're looking at and what kind of groupings you're looking at, it might require you to split these 7 groups into kind of subgroups. And so that has resulted in up to 30 different taxa being recognized by taxonomists today, which is quite a few. I don't want you or need you to learn all 30 of these taxa. Just know these main 7 and their order and what it means as you go down this classification structure. So the most specific grouping are the species, and each species gets a name, which is from binomial nomenclature, right? So you get a genus species name. The genus name is always capitalized and the species name is always lowercase, and both the genus species are in italics and they're written in a Latinized format. So lately we've gotten a little bit creative with the Latinization, but essentially they're in Latin. some sort of Latinized word. A genus is very unique to a very specific group. You cannot have the same genus name for different groupings that are not like they're not related to each other. So genus, you can't have a genus and then this organism was way distantly related also has the same genus name. It has to be unique to that one particular grouping. But you can have the same species name within multiple genus. So you can have, let's say, this genus of organism has one species within it that's their species name is Californica. You can have another genus that has a species within it that has a species named Californica. So that's totally fine, but you cannot reuse the same genus name. And as I mentioned before, this system of classification was morphologically based. Do you share similar physical characteristics with something else? But now we've moved into using this system with the basis of evolutionary relationships and evolutionary descent. Okay, so now you're like, I get it. Kingdom, phylum, class, order, family, genus, species. We got it. But what is the actual species, right? So you've heard me say this word many, many times. You probably heard it at throughout your studies here at GSU and at other schools and high school. You've heard it on TV. You've heard it everywhere, right? But what does it actually mean? Well, species is actually difficult to define. It does have three common characteristics that are shared amongst the various definitions. So different scientists have different ways of classifying what a species is, but these three characteristics are shared no matter what your definition of a species is. All the organisms within a species must have common descent, so they must all come from a common ancestor. A species must be the smallest distinct grouping, so it can't be multiple groups within. It has to be the smallest. You can't get really any smaller than the species. And then it must be a reproductive community. So the organisms within a species must be able to reproduce in nature and produce fertile offspring. Every species has a evolutionary duration. So no species lasts forever. We also will not last forever. Every species has a particular time within the greater picture of evolutionary history where they exist. They also have a geographic range. So a species can be cosmopolitan, which means that they are found everywhere. A good example of that are humans, right? We're cosmopolitan. You can find us pretty much everywhere except for the Arctic. Or yeah, a species can be endemic. So a species that's endemic is only found really in a select area. A good example of this are kangaroos. Unless you're like at a zoo or something, you wouldn't expect to find a kangaroo other than Australia. If you saw a kangaroo in the wild in Jersey, you'd be like, what is going on, right? So they're endemic to Australia. They have a specific area where they live. A species can also be continuous in its range or disjunct. So an example here in this image is showing you that this species that would be distribution, which would be represented in green, is continuous. It flows all the way through Canada and Alaska in one continuous kind of spans, right? But if you're a disjunct species, then you might have a couple of populations, right? But they're separated by an area where there are none of that species present. So this kind of two distinct populations of the same species that are separated by a space where there is no members of that species are present. And the geographic range of a species can change over time. Life is dynamic, the world is dynamic, habitats are dynamic. So the ranges of that species or the characteristics of that species will change over time. It's not something that's fixed. And an interesting concept that I think it's important for you guys to know about is DNA barcoding. So genetic analysis has allowed us to be able to use molecular techniques to identify different species. So this can be argued to be an even more accurate or even more precise way of identifying different species. And to do this, we look at the mitochondrial cytochrome C oxidase gene because within the same species, the gene sequence of this mitochondrial cytochrome C oxidase is relatively conserved. There might be small changes here and there, but within the same species, the sequence is pretty heavily maintained. However, the sequence of this gene between two species is vastly more different, right? So if you look at compare the gene sequences of this gene between different, what you think may be two different species, you might find out that these two organisms that you thought were different species are actually the same species, or you might find that they're actually different species using this genetic analysis technique. So now that we kind of understand what a species is and we kind of got a general idea of what it is, how do we compare one species to another to find out how they're related? How do we find out the relationship between the various species on Earth? So one way we represent this is through using phylogenetic trees. And phylogenetic trees show you the relationship between species that are both extinct and extant, right? So extant mean are living currently, extinct, they've been wiped out, right? And so something to be very aware of is that evolutionary trees show you relationships and relatedness among different species. It does not show you superiority. And so this is a common misconception when people are looking at phylogenetic trees, depending on how they're written. Sometimes people will look at them and infer that an organism that's higher up or an organism that's further to the right or left is superior to another one. That is not the case. That is not what a phylogenetic tree shows you. It's just showing you the relationship that different species have with one another. Okay, so just keep that in mind. The tips of a phylogenetic tree represent different species. So here, A, B, C, and D, and E are all different species. And then you have your nodes, and I'm going to draw dots because, at least for me personally, when I draw dots, it helps me to kind of follow the trees better. And the dots, the points where you have branching are termed the nodes. Okay, and these nodes represent a common shared ancestor, and this also represents a speciation event. So this is when this particular common shared ancestor, this species, underwent a speciation event to result in two separate species. So as an example here, this green dot is the recent common ancestor of A and B. And it underwent a speciation event in order to give you the two different species, A and B. Okay. And then we have our clades. And a clade is a group of organisms that is the most recent common ancestor and all of its descendants. So this example here, it includes C, D, and their common ancestor, right? If you wanted to draw a even larger clade, you can draw a clade that looks like this. This would also be a clade, and it would be the clade that includes species A, B, C, and D, their most recent common ancestor, and then the ancestor of their ancestors, right? So it would include A, B, C, and D, this guy here, this one, this species, and then this one here. Okay. Something to also to note are sister groups. So sister groups are two groups that are share a common ancestor and they're most related to one another. So A and B in this case, A and B are sister groups to each other. They share a common ancestor, which is the green, the species represented by that green dot there. And they're most closely related to one another. You can also have A and B is a sister group to C and D, and they share the common ancestor that is represented by the orange dot. So sister groups are important when you're looking at phylogenetic relationships among species to kind of compare their characteristics. So the phylogenetic tree is based on synapomorphy, and these are characteristics that are shared characteristics due to a common ancestor. So these are homologous characteristics, and these can be morphological, so they can be physical characteristics. They can be chromosomal. They can be molecular. There are a variety of ways to kind of look at these synapomorphies, and this is looking at an ancestor and seeing if, from this ancestor, do they have similar traits? So if we lose our example here, and this is getting a bit crowded, so let me do some erasing. So if we had, let's say, spinal cords were first recognized in this ancestor here, this orange ancestor, and all the descendants A, B, C, and D have a spinal cord, so we know they share that common ancestor. But let's say in this common ancestor is when we first saw hair develop or something, right? So C and D have hair, so hair is a synapomorphy of the clade that includes C and D, whereas the clade that includes A, B, C, and D and all of its ancestors, so like this clade, the synapomorphy of that clade would be having a spinal cord, right? So it's the common trait, common characteristic that is derived from an ancestor. So all the descendants have that trait. So when we're looking at our phylogenetic trees, there can be different groupings. And the first grouping is a monophyletic group. So you heard me use the term clade. The term monophyletic is synonymous with the word clade. And to reiterate, it includes the ancestor and all of its descendants. So when you're looking at this blue area in this image here, we have our common ancestor, right? And all the descendants. So we have this descendant, and all of these descendants. This is a monophyletic group. They're all included. Then you can have a paraphyletic grouping, and this is where you include a recent common ancestor, but not all of the descendants. And so in this image here, if you look at the blue area, there you have a common ancestor, and we have several other common ancestors being included. And we have C, D, E, F, G, and H included, but this grouping here of A and B have been excluded. And this makes this grouping paraphyletic because it does not include some of the ancestors that derived from that, some of the descendants that derived from that shared common ancestor. And then we can have polyphyletic groupings. And this is where you include different species, but they do not, you include related species, but you do not include their ancestor. And so in this case, you can see how they've included C through G in this grouping, but have not included this or this or this common related ancestor, right? So they're missing some of the ancestry here. And we'll talk a little bit more about some of these groupings and are they considered valid or not in a couple of episodes. Sorry, not episodes, I mean slides. So we're going to talk about some of the techniques, the taxonomic techniques that are being used and today, and how we end up with these paraphyletic and monophyletic and polyphyletic groups. And so first we're going to discuss the evolutionary taxonomy method. And so if you're an evolutionary taxonomist, your method of classifying and the relatedness of different species is based in Linnaean taxonomy. And when you're kind of going through and putting your trees together, your groupings together, you're looking for a couple of different criteria. All the taxa, so remember a taxa is a group of, just a group of organisms. All the taxa must have a single evolutionary origin. It must include all members of a taxon and must be convex. And the base of this evolutionary taxonomy classification is based on adaptive zones, and the adaptive zones are just the unique a group's unique way of life. So we're going to use this this phylogenetic tree of primates as an example. So if you're an evolutionary taxonomist, you look at this tree and you're like, okay, this tree is valid. You know, ignore the top designations. You're just looking at the blue bar, the blue branching. The branching looks good. They all these organisms have a single evolutionary origin. All the members of the taxon are present. It's convex. It looks great. So you start to try to put these organisms into different groups. Well, if you're an evolutionary taxonomist, you include humans and their most recent common ancestors in Hominidae. Okay, and then you include the chimpanzees, gorillas, and orangutans under Pongidae. And so you do this because you're looking at their adaptive zones. When you're looking at humans, chimpanzees, gorillas, and orangutans, well, you notice that chimpanzees, gorillas, and orangutans are all arboreal, They are herbivorous and they don't have a language, if you will. Okay, so you're like, okay, those they have similar adaptive zones. They have similar ways of life, so they're grouped together. And then humans, we don't get grouped with them because we are terrestrial. We live on land. We're omnivorous, and we have culture and language, so we're grouped separately. But if you've been paying attention, you might have noticed something interesting. Why is it that humans are not included in this group with chimpanzees? Because we have a similar common ancestor. Chimpanzees are closer related to humans than they are to gorillas because we share a closer common related ancestor with chimpanzees than chimpanzees do with gorillas. But chimpanzees and gorillas are grouped the same if you're an evolutionary taxonomist. This causes you to get a paraphyletic grouping. So Pongidae, if you're an evolutionary taxonomist, is a paraphyletic group because it does not include humans, right? So a lot of taxonomists had issues with this. They're like, we don't like this paraphyletic grouping. Humans should be included with any group that has chimps because we have a shared common ancestor. Why aren't we being included? And so a different method was developed. Let me erase here. There we go. So instead of using the evolutionary taxonomy method, some people have switched to using the cladistic systematics method, and this system is all based on common descent. So common descent is key, and in that case, if you're a cladistics uh, taxonomist, you include humans in with the same group as chimpanzees, gorillas, and orangutans because now this is a monophyletic group, right? It includes common ancestors and all of the descendants. Whereas when we were looking at it from an evolutionary taxonomist perspective, it excluded humans and gave us a paraphyletic grouping. So in the cladistics form of taxonomy, monophyletic groups are the only valid groups. So today we use a kind of a combination of both, heavily leaning more into the cladistics area of things because we want monophyletic groups. Monophyletic groups are what we want to have the most accurate depiction of the relatedness between species. So, but we do use a little bit of both of these methods nowadays. So just as a overview, going from right to left, if you're an evolutionary taxonomist or a cladistic taxonomist, you do not recognize polyphyletic groups as valid because they break the first rule of evolutionary relatedness, right? If you're an evolutionary taxonomist, they have to be all share a common ancestor in that grouping. And same thing with the cladistics. And in polyphyletic groups, you're excluding those common ancestors, so it's not valid. If you're looking at paraphyletic groupings, if you're an evolutionary taxonomist, there are some paraphyletic groupings that they consider to be valid, but if you're a cladistics taxonomist, no, they are not considered valid because they do not include all of the descendants, and so you're now messing up the monophyletic grouping that could occur. And then we have our gold standard, right? So monophyletic groups. That's the ultimate goal of what we want in taxonomy, no matter what your system is, whatever technique you use. And so as we're going through time, taxonomy is growing and changing in an effort to get all of, if we can, all of the diversity of life into distinct monophyletic groupings. Okay, so that kind of concludes the basis of what I want you guys to know on taxonomy. Um, and then we're going to start going from this point into looking at the individual phyla of life. There are 32 monophyletic phyla of life on Earth, right? So if you're looking at this image here, all of the um, all of the organisms included under Animalia are animals, right? So it's quite a bit, which is great, lots of diversity. And they're split into taxons as you kind of go through the tree. And so some of the taxons you see here are like Bilateria, Protostomia, Deuterostomia. Ecdysozoa, Lophotrochozoa. Some of these terms, many of these terms you should be familiar with because we've already discussed them in previous lectures. Some of them you might not be, but these are kind of like some of the taxonomic groups, and they're based on the synapomorphic characteristics, right? So the blastula, the presence of a blastula, if you're diploblastic or triploblastic, um, the spiral cleavage, deriving from a common shared ancestor. The mouth is formed from the blastopore, which is a protostome, right? Derived common ancestor. So just familiarize yourself with this tree. You don't have to memorize it or anything at this point. As we're going through the semester, we're gonna uh, hit on these 9 major phyla, so Porifera, Cnidaria, Platyhelminthes, Nematoda, Mollusca, Annelida, Arthropoda, Echinodermata, and Chordata. So as we're going through those, this tree will kind of build and build and build in your mind and make more and more sense. So just get yourself familiar with it. And as we're going through, we'll keep revisiting this phylogenetic tree. So some key points to take away from this lecture are: what are homologous and analogous traits? What does it mean for a trait to be a characteristic to be homologous versus analogous? What are taxonomic groups, right? So what is the hierarchy of the taxonomic groups? What are the names? Like, how do you order them? And then binomial nomenclature, knowing what that is. Know the general characteristics that define a species, know the purpose of a phylogenetic tree and what they can tell us about the relationships between species. Know how to interpret a phylogenetic tree, so know how to read it and what does it mean, and when you see 2 species on a tree, what does it mean when they're grouped in different ways. Know what a synapomorphy is, and their use and how they're used in phylogenetic analysis. Know the difference between a monophyletic, paraphyletic, and polyphyletic group. And then familiarize yourself with the terminology that I mentioned in the last lecture. So the lecture covering chapter 9, go back to the slide that is slide 8 and make sure you know all the terms on that list, because those are synapomorphies that will come up throughout this entire semester. So make sure that you know the definition of all of those terms. All right, we've reached the end of this chapter. This is the last chapter you'll have before your exam, so be sure to read chapter 10. I've picked out certain sections of chapter 10 that I want you to focus on, because there's a lot of detail and it's all very important, but there's certain things that I think are kind of important to get the big picture. So read sections 10.1, 10.2. In 10.2, just pages 204 and 205, and then the barcoding section of page 208. And then read section 10.3, and then page 211 for 10.4, and then 10.5. Then I've also included some videos here that I thought were really, really helpful. The first one is actually my own video from 1104, and that's really for if you're struggling with some of the terms that I used in this presentation, like speciation event and things like that. I would go ahead and just watch that video that I posted on the origins of diversification of life, to get familiar, to remind yourself about some of that terminology. There's also the crash course video on taxonomy, which is really good. I'm a big crash course fan if you haven't been able to figure out yet. Biology Makes Sense. They made a video that's really good at explaining the taxonomy, and same thing with the difference between phylogenetic trees and how to read phylogenetic trees. So if you need more resources to understand this material, or if you're just interested in what these videos have to bring, I would say go ahead and watch those. So excellent. I hope all your studying is going well for the exam. Uh, just make sure you study hard. If you have any questions, make sure you're coming to office hours to ask, or going to the tutoring center, or talking to your peers about those things. So excellent. I'll see you soon.